Hi everyone, we're going to continue our lesson about the human genome, and we're going to start about identifying genes. So in the last lesson we talked about DNA fingerprinting. So once we have the sequence, once we've got the genome sequence, the next step in the process is to identify the genes and to determine their functions. So you want to identify the gene and you want to determine their functions. So the functions of many of the genes in the human genome are still unknown. So we don't know most of them, right? There's so many genes that in the human genome that we don't know the function of. Now, researchers use techniques that integrate computer analysis and recombinant DNA to determine the function of these genes. So how do researchers identify genes and their functions? Well, they do that by using computer analysis and recombinant DNA technology. Cool. Now, when we talk about bacteria and yeast, their genome does not have large regions of non-coding DNA. They do not have large regions of non-coding DNA. So researchers have identified genes by scanning the sequence for open reading frames. What are open reading frames? Open reading frames are stretches of DNA containing at least 100 codons that begin with a start codon and end with a stop codon. There isn't anything else that you should know about identifying genes at this open reading frames at this level. The only thing that you should be aware of is that they are stretches of DNA. They should contain at least 100 codons, and if we remember, a codon is three bases that code for an amino acid, right? So we need to have at least 100 codons that begin with a start codon. So if you remember the start codon AUG, right? So it needs to begin with a start codon and it needs to end with a stop codon. And the stop codons include the UAA, UGA, UAG, right? So what are open reading frames? They are stretches of DNA containing what? At least 100 codons. They need to start with a start codon and they need to end with a stop codon. Right, while these sequences might indicate a gene, they will be tested to determine if the sequence produces functioning proteins. So while these sequences, they might indicate a gene, so they might be a gene, we test them to determine if they provide functioning proteins, right? So we test these genes to check if they have, if they code for functioning proteins. Right, so we talked about that a codon is a group of 3 nucleotides and that they code for an amino acid. And the start codon is AUG and the stop codons are UAA, UGA, and UAG. Now, open reading frames analysis has been used to identify correctly over 90% of genes in what? In yeast and bacteria. So this open reading frame has been used to correctly identify 90% of genes in yeast and bacteria. However, the identification of genes is more complex, and more complex organisms, such as humans, requires more sophisticated computer programs called algorithms. So to identify the genes of more complex organisms like us humans, we need more advanced computer programs, and those are algorithms. So what happens is these algorithms will use information such as what? The sequence of the genome of other organisms to identify human genes. So identifying human genes is not an easy task, right? It's not as easy as identifying the genes for yeast and bacteria. Now, let's talk about bioinformatics. So, the completion of the Human Genome Project and the sequencing of the genome of other organisms have resulted in large amounts of data. So, once we finished the Human Genome once we finished the Human Genome Project and we sequenced the genomes of other organisms, now we have large amounts of data. And not only has this enormous amount of data required careful storage, right? We don't want to lose the data that we've collected. We've worked on years, spent a lot of money trying to find these sequences. Like, we can't lose them, right? So we need very careful storage, organization, and indexing of sequence information. We need an index, right, to know which one is for what. But it also has created a new field of study, and this field of study is called bioinformatics, and it involves creating and maintaining database for biological information. Basically, if you break the word down, bio basically means we're talking about biology, right? Biological, and then informatics is information. So this is a field of study where we create and maintain databases for our biological information. The analysis of sequence information involves finding genes in DNA sequences of various organisms. So, what does the analysis of sequence information involve? It involves finding genes in DNA sequences of various organisms and developing methods to predict the structure and function. So we're predicting what? We're predicting what? The structure and function of what? Of newly discovered proteins. So let's read that again. The analysis of sequence information, so us analyzing sequence information, it involves finding genes in DNA sequences of various organisms. So not just humans, we find genes in DNA sequences for various organisms, and we develop methods to predict the structure and function of newly discovered proteins. So we use this information, this analysis of these sequences that we've got to predict structure and function of newly discovered proteins. Now, scientists also study the evolution of genes. So they also look at the evolution of genes by, how do they do that? So we know that they study the evolution of genes, right? How do they do that? They do that by grouping protein sequences into families. What does that mean? What does grouping protein sequences into families mean? It means that we take proteins that have related sequences, right? And we compare them to similar proteins from different organisms. Well, I still don't get what that means. Well, basically what we do is we group protein sequences. We take sequences that are coding for protein and we put them into families. What are these families? These families are related sequences. So these proteins have similar sequences, right? And we compare them. We compare similar proteins from different organisms. So we group similar sequences and we compare similar proteins from different organisms. So if you have a protein that is similar in humans and in other mammals and in bacteria, so we take that and we compare it between those different organisms, okay? We'll talk about DNA microarrays in the next lesson. See you later. Bye.